Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I have a few announcements to make. We are launching our GPCR consulting services. More information will be available soon. In the meantime, if you need support with your R&D project, understanding the pharmacology of your receptor, or need help getting an idea off the ground, email us at hello@drgpcr.com. We have fantastic talks coming up at the Dr. GPCR Virtual Cafe. Visit drgpcr.com/virtual-cafe to see our upcoming live events. Get your tickets today on Eventbrite. Don't forget, space is limited. To become a speaker or to sponsor an event, you can fill out the form on the main page of the Virtual Cafe. Go to drgpcr.com/virtual-cafe today. Are you subscribed to our monthly newsletter? Stay on top of all your GPCR news by subscribing today at drgpcr.com/newsletter. Also, to watch previous episodes of the Dr. GPCR Virtual Cafe or the podcast episodes, subscribe to our YouTube channel today. Last but not least, stay tuned for more information about the upcoming Dr. GPCR Summit held between September 13th and 19th, 2021. Visit drgpcr.com/summit for more information and how to submit your talk. And now, let's dive in. Hello, everyone. This is Yamina from Dr. GPCR, and I have the great pleasure of talking to Dr. Brian Arey. This is part 2 of our conversation. We had so much fun, Brian. Thanks again for coming back.
Thanks for the invitation. It's exciting. The podcast is incredible. I really enjoyed watching them. So congratulations to you on a great job.
Thank you. Thank you so much. I really enjoy it. And it cannot be as good as the guests that come on board. So last time we talked a lot about your career, the book that you wrote, which I haven't had a chance to look up, but I will, I promise. And so as everyone knows by now, at least those who have had listened to episode 1 of our discussion, you're a scientist in industry.
Yep.
And just a real quick, idea on where you started. If I remember correctly, you started out in academia, and at some point you had the opportunity to go into industry, and it was you never looked back.
That's basically the story. Yes. Well, I mean, there was a choice to make, and you came to a crossroad. So it was not an easy decision, as we talked about. There was a lot of angst, but in the end, you don't know when the next opportunity is going to come, so you have to take the one in front of you.
Exactly. And if I remember correctly, you have a long arm long worth of titles, and you're leading a team of about 70 people.
Yeah, in an interim basis, just to be clear. But still, it's been a lot of fun. I've really enjoyed a lot of it. So.
I know we talked about this a little bit, but can you remind us how do you manage those 70 people? Is there a hierarchy? How do you, because in my mind, when you say 70 people, it's like you're up 24/7.
No. So, I mean, I can understand how somebody might think that, but no, there's certainly a hierarchy. And as with anything in industry, there's a hierarchy of management, and supervision. So, I report into people up into the CEO. If you looked at the ladder, I ultimately report into the CEO, but there's a gradations of supervisory management all the way through. And so I have at this point in time 10 direct reports. So those are the people that I actually supervise on a daily basis. My job really in this interim role has been to drive and try to maintain the organization functioning properly and efficiently, but also looking toward the future and developing strategy along with those guys. So it's not something that I just sit in a room and I come up with a strategy and I say, this is what we're going to do. Really, those guys that are on the at the bench or leading the labs are the people that really know better even than I do, right? So you have to rely on them and their knowledge. So it's definitely a team effort. I'm ultimately responsible, but those guys are so good, and a lot of them are really, really experienced. I mean, they have 20 years or more in some cases of experience in industry, so they know what they're doing. So I tend to try to step in when I'm needed, but not to get in their way. Because, like I said, and especially in the interim role, I mean, I basically have 3 groups. I have the mechanistic pharmacology group, which really was the group that I started, that I know a lot about, and I'm a little bit more hands-on there. But the compound management and the coordination groups were really already really well established, and I just got this opportunity because somebody left, and they needed somebody to take on the interim leadership. But at the same time, you try to have your own impact, right? It's just not like, right. So that will be ending soon, probably in the next month or so. So, but I'll miss it.
That's so interesting, because we talked a little bit last time as well about your leadership style and the importance of communication. And before we hit record, we talked a little bit about year-end evaluations. And what are your thoughts about communicating with your team, and how do you do that with your team on a regular basis?
Well, I have weekly meetings. I also have monthly, what I call one-on-ones, if you will, where we just sit down for an hour or so and talk about issues or whatever. But the industry is such a dynamic environment, and when you're in when you're running support teams like I'm running, that sort of reach across the entire preclinical research end of a company like this, it's $130 billion, whatever. It's huge, right? It's a lot of stuff. It's really almost on an hourly basis. I mean, seriously, I get emails constantly, and you have to address them in real time as best you can. That's the part I really like about it. I like that excitement. I get bored easily. And so I don't like being bored. I've been in those situations before where I've sort of been languishing and kind of bored, and I never wanted to get back there, because it's really that's when you really have an impact, when you're in the mix, and it can be frustrating and stressful, but at the same time, it's a lot of fun. So yeah, that's really how you do it. I mean, there's so much going on, and in terms of communication, I always like to keep things fun. That's something that I've sort of always had about me when I was at work. Even when I first started doing just my first job, I always wanted to have fun while I was doing it, and so I try to keep it fun and relaxed as possible. That doesn't mean there's not times to be serious, but if people are having a good time and they feel like you're approachable and they trust you, they're going to get the best. That's always been my overarching philosophy.
I think that resonates a lot with a lot of posts on LinkedIn and a lot of books that come out that that basically tell you when an employee feels valued, when they feel supported, when they feel like they have what they need to do their work, they're going to give you 150% of that energy. Because
it's about trust, right? So it becomes about trust, and it also becomes about, if they feel that you have their best interest at heart, which I do sincerely, and they understand that sincerity, they will follow you anywhere. They will. And I posted something on LinkedIn yesterday about this because I feel so strongly. But I just happened across. I'm like, yes, this is exactly.
I saw it. I saw it, and I was reading about it, and I said, well, yeah, I think we're all on the same page at this point when it comes to this. This is the Dr. GPCR podcast, and last time we didn't get a chance to talk too much about GPCRs. You had mentioned that you're still working on GPCRs. And but then it's a secret, so we wouldn't be able to publish the episode. But since you're in industry, I wanted to ask you, can you tell us a little bit, kind of in a step-by-step manner, on how some of these projects that you worked on were initiated and how they went through all the different stages?
Yeah, so that's really. This is, again, everybody's going to approach it differently. And even you'll probably find different pharmaceutical companies have different approaches to doing this. But it really all starts with the disease that you're trying to target. And you start at that point, right? And understanding pathophysiology and mechanisms of pathophysiology. And once you can develop hypotheses by studying the pathophysiology or the mechanisms of pathophysiology. And from there, so as a physiologist, that always speaks to me, right? As a physiologist, trained physiologist, and that's why I'm most comfortable. But then trying to reduce that to practice. Okay, what does that mean for the cell? What does that mean for the system? What does that mean for interacting with other systems? This is how some of the I'll give you an example. So this has already been published. So at least in presentation format. I got into endothelial function and trying to understand how endothelial cells lead to cardiovascular disease and what their role was. And again, that was a part of taking a path less taken. So I think I explained this last time I mentioned it, where I went to my boss and I'm in this new area and I really didn't have a whole lot of understanding about it. And I asked him, where's the opportunity? And he said, well, nobody's working on this. So, okay, let's go work on endothelial cells. And out of that, came a really fascinating research program looking at mechanotransduction and understanding how GPCRs could function as mechanotransducers before anybody else was even thinking it that way. And so you start there, and then you have to validate your hypotheses, right? So you have to make sure that there are tools available. For example, that's a positive. It's harder if you're really breaking the validation of the pathophysiology. So meaning, for example, when I got into the endothelial cell work, it was already well known that endothelial cells respond to flow, the mechanical shear force, and what that meant for atherosclerosis. So that was well established. They just didn't understand how it was responding. So that was a prime opportunity to step in and say, okay, maybe we can figure this out a little different way. But that's really at the heart of it. That's where you start. And then you try to relate that to animal models, of course, and ultimately the human condition. And then once you've got to that point, then you can really start to hone in on targets that might modulate that system. There's a lot of people who think that because you have a micromolar compound, you have a drug.
Well, that was my next question. Is how do you select the best target? And also, when do you say, well, this molecule works?
Yeah. So how do you pick the best target? So that's really, that's a real personal thing. But in drug discovery in the modern era, these are team efforts. These are not done in isolation. So, for example, you'll have input from other scientists within your department. So in the case of cardiovascular disease, for example, you get input and advice from people, and you try to build through your data, you try to build consensus that this is a good project. In some ways, it's like trying to convince a grant committee that your grant is worthwhile funding, and you just do it in a different way. And so there is that aspect, and parallel to academia, where you have to build consensus that your project is both of value as a potential medicine, but but also of value from the perspective that it's an investment that the company wants to make and will get something back from. So, and that's, that takes a long time to learn. But as with everything, the more you talk to other people and the more input you get from other people, you learn the skill.
That's such an interesting, it's so interesting what you're saying, because you can very easily draw a parallel about drug discovery and this process that you just described. And as you mentioned in academia, getting the grants and then getting support. And also you need results at the end of that period, which will get you more funds. And obviously in industry, the whole goal is to get a drug out there, because it's still a business. So you need a drug to go back and start again with different projects.
Well, I think, I want to stop you there, because I think you said something, it's a business, but it's also about helping patients. And I think that's a critical aspect that people often don't understand. A drug discovery scientist doesn't go into the lab every day thinking, wow, I'm going to just make money for the company. That's not their goal at all. I mean, and in fact, the vast majority of people go in really, truly wanting to help patients and to solve problems. And so I think that's really important. And you can see that. I think, Michel Bouvier is just a prime example of how to do this in a balanced way. The academic part and the industrial part, I really admire his ability to sort of span both worlds. And as I said the last time, he's kind of a mentor. I kind of, even if he doesn't know it, I kind of try to emulate him in a lot of things that he does. But
agreed. I think the way I see the money or the cash intake from anything, it's more like a fuel. So you have a car.
Correct.
The car is the industry, whatever you want. But at the end of the day, you need that fuel in order to realize your mission or to get where you want to go.
Yeah, and early discovery, what we call exploratory work, is a difficult place to live. And I know because I lived there for a long part of my career. And it's because you get little recognition. Most of your ideas don't work. And you spend a lot of time just turning the wheel, trying to find the target that's the right one. In case of the endothelial cell work that I'm currently writing up or trying to, in between my day job and my life, that was a prime example. It was very exciting where we're able to find some really, really good targets. And at the same time, break ground that hadn't been done before. So that was kind of like the perfect sweet spot of science where you're able to do new things and at the same time develop some new drug discovery targets. What was I going to say? Yeah, I mean, I think so. You have that early exploratory spot. It's tough, but you have to be, you have to have faith in your own ability and get help wherever you can. Really, from other people, from academic sources, from other people in your own team, etc.
It's interesting because I think the early discovery part of drug discovery sounds a lot like academia, where you really have the opportunity to be creative. You hope for the best. And if, as you mentioned, you find that sweet spot where you're discovering something new, you get a molecule, you get a drug target that is worth pursuing. And then you have all the data together. That's the best way. But then oftentimes experiments don't work, or the company changes their idea and says, well,
oh yeah, that happens.
Although this is interesting, but we are not focusing on this anymore.
Yeah, there's a concept of industrial or what's the word I'm looking for? Organizational patience. That's what I call it. And they have a certain level of patience. And sometimes nowadays, I think it's a little bit longer than it has been when I was in the meat of my career. But certainly there's a lot of pressure around that, right? To churn out a target or get things moving along. Because, in the end, there are other targets out there that they want to prosecute and they want to work on. And if you're taking up resource to do that on things that they don't have a lot of confidence in, they're not going to stick around for that for too long. So you really have to show progress at all stages. And that's how the other thing you picked up on was getting more and more resource over time. And that's absolutely true, right? So in order to prosecute a drug discovery target from the early exploratory stages into what we call full phase drug discovery, you increase incrementally the support that you get from across the organization. So for example, chemistry or ADME help and things like that. And that all comes into play into these large, when you get to a full phase program, you get into this large program that can be 30 people. We're all working on the same thing.
Which I think must be very rewarding when you start out with an idea and you talk to a couple of people and then you get to that point where you acquired enough data, but then you rallied 30 people around it and full funding and full support from the company. I think it sounds to me that it's a very rewarding point to get to. Yeah,
but then the pressure gets ratcheted up.
It's with everything.
There's a price for everything, but no, it's really true because you're taking up when you've reached a full phase recognition or governance, what we call it, so we have to present these programs and their progress over time to various governance committees, and then they will sign off on getting more support or whatever. So again, it's sort of like constantly looking for new funding. And when you do that, then that comes with an expectation. And so, there are current programs that I've been involved with for many years that I started that I'm sort of loosely associated with now that they've been around for a long time. It's time to, it's trying to put up, as they say, up or shut up.
Yeah. Yeah. It must be, I can only imagine the stress level of that increases over time whenever a program gets more attention, more funding, more development. But then again, I think it goes with the excitement of getting into a clinical trial and, getting into.
Oh yeah. That's the overarching goal for every one of us, right? We want to get and say, okay, we, got that drug target the clinic and let's see how it works. And yeah, I mean, that's the holy grail. The holy grail is sort of working on compounds that actually make it into the human and treat disease. But even that of getting something across to the clinical side, which gets more and more difficult because nowadays the hurdles of safety are much greater than they were, say, when I first started, and rightfully so. You really have to make sure you have something that's really solid before they're going to invest in it.
Of course. And you don't want to jeopardize anyone, patient's health.
Of course.
And you really want to make sure that whatever.
Oh, yeah, that too.
At the end of the day, I think that's the whole point. If you have a molecule, although it may sound, beautiful in cells or in mouse models or, any animal models, but at the end of the day, you want to make sure that, it works great in humans with limited side effects.
Well, I think what's really kind of funny is over time, you become if you work on a project long enough from its inception up to the point where you're getting into the full phase drug discovery effort, especially if it's cutting edge stuff like what we're doing with endothelial, you become probably a world leading expert by that time. I mean, because you're constantly doing basic research to try to understand the system better. So there was a time, I think I probably knew more about the KLF2 pathway and how it affected endothelial function than anybody else on this planet, because I mean, we had done so much legwork to try to show that was a valuable pathway. That it was we knew all different kinds of inputs that ultimately I watched get published by others, but it's validation all at the same time, right? That you're on the right path.
Yeah, I think it must be, although it's not you who published it, but it's such a great feeling when you read an article and you say, aha, I knew it. I knew this is how it was supposed to work.
Yeah, it can be frustrating too. That's the toughest part. That's the toughest part. We're all scientists and we're all trained in the same system.
Yeah.
And so, that can be tough to deal with sometimes. But at the same time, again, you got to keep the eye on the prize, which is you're trying to treat disease, and that's your higher goal.
Exactly. Before we hit record, we were a little bit talking about, taking a path less traveled and asking the questions in science that may not be the flavor of the year or of the month. But I think as you just mentioned, seeing these discoveries being made and published by others, and that they're right because you've already gone through the whole process. Yes, it can be frustrated, but at the end of the day, as you mentioned, it's a discovery path, and you need to keep your eye on the prize, as you mentioned. But also still, when you ask those, out of the blue questions that you think are important, and then it takes some time for people to realize, well, actually, it is important. Yeah. And you go home and you say, oh, I had the right idea from the beginning.
Right. Well, there was a paper that came out a couple of years ago on GPR56, right? Mm-hmm. Showing that GPR56 was a mechanosensor in endothelial cells, and literally I knew that about 10 years before everybody, and that's just the way it goes. You kind of, but at the same time, it's not unlike parenting in some ways. Parenting can be frustrating in the heat of the moment, but at the same time, you keep your eye on the prize, which is developing and creating this relationship and healthy young adults, and that's the important thing.
Exactly. I think you draw. I love the parallel when I think about my 5-year-old. In my mind, what I want is make sure that he grows up healthy, he can think on his feet, and he can make his decisions, and really be able to stand on his feet and be a strong person.
Yeah.
Take the right decisions that make sense to him, but at the same time be able to logically think about things.
Yeah, you can't. You want your children to be independent thinkers. That's at least I did, and I got my kids into a lot of trouble at school sometimes because they questioned everything. This is another topic we talked on before we hit record. You question everything. I didn't really have a problem with them because I always want them to question and I want them to, draw their own conclusions. I mean, they don't have the same philosophies or perspectives on certain things that I do. And I don't want them to, I want them to have their own thoughts.
I think at the end of the day, if you can have, an intellectual discussion with your kids, mm-hmm. on a topic on which you don't agree, but you still can defend everyone can defend their own opinion. I think it's just the most rewarding thing ever.
It's absolutely rewarding. Absolutely. That's how you know you've done a good job.
I know. I have a 5-year-old, and just a funny thing. So they were on Zoom last week because they closed the school for the 3 days, and the teacher kept asking him, "So Attila, what is the name of this? Can you answer me?" And he responded very sincerely, "No, I'm hungry." And I thought that was such a direct and sincere answer. I can't answer because I'm hungry.
I'm done. Right now, I'm done.
And the teacher took it so well. She said, "Well, I think I understand you. It's almost lunchtime."
That's good. But I mean, to circle back to your other point about, what makes a good compound,
Yeah.
There's a lot of things that go into that. And I think that, that's another art. But this is where I have to say, no one person can know it all, and no one person can really have the entire perspective of what makes a good compound. Obviously, there's a lot of things that go into it. It has to have the right pharmacokinetics. It has to have the right, pharmacology. It has to have the right distribution. It has to have, there's so many different aspects of it. Its half-life has to be ideal. You have to have an understanding of how you're going to administer it in order to get the desired therapeutic effect. Yeah. So that's why you have 20, 30 people working on it from different fields, because those people have their expertise and they can tell you. I am sincerely amazed with what we can do with chemistry today. It's mind-boggling.
I think it makes a lot of sense that you need that team effort. And even in academia, we talk a lot about teamwork, and in industry, based on my limited experience, teamwork has a different meaning. But again, the goal is very different. But yes, having 20, 30 people who are all experts in their own field, and putting everyone in the same room and saying, well, this is our target, this is our problem, how do we get there? And then getting that, distilling down that path where everyone, for the most part, agrees, because everyone has that ability to add in that input. I think it's just phenomenal. And as you mentioned, getting to a drug is not an isolated effort of one person.
And this gets the whole concept of intellectual humility, which I'm also a big proponent of, which means that if you're leading these large drug discovery teams, it's never about who has the right answer. It shouldn't be. And there are people that will try to drive it that way, but it's not really. I don't think that's the most successful approach. It has to come from a place where we're all working as a team, on a problem, and we have to find the best solution. And people shouldn't get upset if their solution is the one that works, or selected. And I will have to say that oftentimes mine are not. but at the same time, what you're doing is you're setting up an environment where people feel comfortable sharing their wild, crazy ideas, and that just might be the one that works. So, that I think that's another part of being a successful drug discovery scientist, is the concept of intellectual humility, and understanding that you don't know everything,
and also being able to say something that doesn't make any sense, not because you need to say something because you believe that it might be the solution, but maybe that will trigger a question for someone else in the team, and that will build upon that. I read somewhere that usually a good leader, a good team leader, usually when you have a meeting, gives the answer or gives their opinion last, because this way everyone in the team gets to say what they want to say, and they're not intimidated by the big boss, who may or may not be right with whatever they're saying.
Well, I'm not really a big boss, but I do tend to try to be as quiet and let people. I always like to create a discussion. I think it's you create solutions through discussion and sharing of ideas, however crazy they might be. And the whole concept of there's no stupid idea is really true. I feel, at least in the teams that I've led over the years. I don't really feel like you should squash ideas because it just might be the one that solves the problem. And this is really what drug discovery becomes over time, is you're just solving problems, right? You identify a lead molecule that came out of a high throughput screen, a certain chemotype, if you will, which has a certain core structure, and then you're trying to manipulate that core structure in order to get the desired PK or the desired ADME profile, while maintaining therapeutic benefit. And as you can imagine, that's kind of a tricky thing to do. So it has to be a discussion. It has to be, people sharing and creating to find solutions, and because that's really what it's all about. You're trying to balance the solution for this problem with the solution for this problem, and it becomes. But it's fun at the same time. It's all fun.
It sounds like an orchestra. You're sitting in the audience and you're listening to classical music and you're watching all these musicians playing their own instrument, and then you're there to harmonize everyone and get something out of it. Which in the case of music, it's a symphony, but in the case of drug discovery, is to get to that molecule, hopefully. At least
I love that. I love that analogy. I don't know if it's actually accurate, but I love it. I think it. Yeah, I think it really is. It's about it. I think it's just about providing a team environment where people are comfortable, and it's not accusatory, because mistakes happen, right? People are going to make mistakes. I don't know if we touched on this before, but the other concept I'm really a big fan of is allowing people the ability to make mistakes without punishing them. Because that's how you innovate, right?
Yes, and that's how you learn. I think finding the way to go from point A to point B is very important. However, figuring out that going through Z before B, that it doesn't work, is also very important. I think the negative data, it's funny because when I was a PhD student, one of my colleagues, at some point we were getting negative data, and we were saying, well, maybe we should found the Journal of Negative Data. And I still think it's a really cool thing, because people spend such a long time working on these projects, and you just show that actually it doesn't work. But that's also very important to know that something doesn't work.
Oh yeah, I absolutely agree. And so you're right. I don't think that's the way that the system is built, because the system is built to encourage people to have new breakthroughs and new things. But you're right. I mean, we can solve a lot of problems by refuting data and saying, no, that's not how it works. And here's as long as you have the reason, right? I mean, if you can't simply say this is not the way it works, but if you have a reason for refuting a certain thought, I think that's a good idea.
Yeah. And then I'm saying, you go through the experiments, you do the right controls, you have a hypothesis, and if the answer is, well, actually, this is not a good hypothesis, it may not have the impact that you wanted it to be. It's not going to be published in Nature, but at least the person who is thinking about the same problem will know, well, these guys did this and it doesn't seem to be working. And now you can actually make the decision as to, do I want to try it, or do I take it for granted that it's not working and I get another idea and work on something or innovate from there?
Wise. So you're very wise, because that's that whole concept really plays into the exploratory phase of drug discovery research, right? When you're looking for new targets, and I talked a little bit about validating targets and creating this data set to convince yourself and others that this target is of value. It's just as important to say, no, that hypothesis is wrong. And the earlier you do that, the better in the drug discovery process, obviously, because if you're garnering resources just for the sake of garnering resources to promote your career or elevate yourself within the organization, ultimately that's going to be discovered. And the other thing is the company's going to waste a lot of money and time, but they could be spent on a target that actually has more value. So that sort of go no go decision is really important.
I think so too, and I think it's a matter of time and energy. When you've spent so much time in the lab, or so much energy or funds on developing assays, and before even thinking about, is this the right assay? Is this the right question? I think it's It's just
I had a supervisor who really, his name is Dietmar Seifert, German guy. We're good friends, and he was, he taught me a lot too. He's like one of the good guys, good supervisors I've had in my career, and because he really taught me the value of that killer experiment. Because he and I were collaborating on the KLF2 sort of mechanotransduction pathway business, and he always insisted on doing really tough killer experiments to make sure that we were honing in on the right things. And I always was very appreciative of that, because it can be hard to make those decisions when you got so many pressures from different things, and that your career progression is built on success. It's an easy thing to fall victim to, and to have somebody like that who really keeps you grounded and as to what's important, and teaches you the value of making those hard decisions and doing those hard experiments is really, really important. So he's basically, I hope he watches, because I admire him tremendously, because he taught me a lot.
It's so interesting. Yes, I think doing the right experiment and then really asking the right question, it can be a time saver, but also it can lead you to discovering something else, realizing, well, this is a good question to work on, or this is not the question you want to work on, because this is not the way to get there. It's kind of like a Google Maps, whether you want to avoid highways or not.
Right, exactly. No, that's right.
To get to your destination. The reason I just thought of this is because I don't like highways for some reason. And every time I go somewhere, I have to make sure that it says avoid highways.
Oh, is that right?
I like the scenic routes, and it's the fall, it's winter, it's not too bad outside. So then why hurry? Why take the highway with a lot of angry people on the road here in the Boston area?
Yeah, the drivers are well known.
It's very different. Have you had the opportunity to go, from an idea and go, follow that molecule or follow that pathway or develop that pathway into a clinical trial?
Well, no, I've been associated with clinical programs, from the research end that went, not ones that I've led per se, mainly because of what we discussed on. There's been a number of things that I've brought to bear that were basically out of scope over time and got shelved. I still feel really strongly about the KLF2 pathway and the GPCRs as a potential treatment for atherosclerosis. But, the fact of the matter is we don't work on that anymore at Bristol. And so we had to put that aside. Hopefully somebody will take that torch up and push it further, because I have a lot of, as an anti-inflammatory pathway, I have a lot of faith in it. And I think it's sort of fallen out of favor. At one time, in 2012, 2014, KLF2 was really, really important in the immune space, and people were really interested in looking at these transcription factors. But it's fallen sort of out of favor with the whole PD-1, PD-L1 thing, and oncology. It kind of got overwhelmed there, and I think, but I think people will eventually get back to it over time, hopefully.
Who knows? Maybe someone listening at this recording will say, "Well, let me."
Well, I mentioned it. let me please do it because it's good. I think that but that's a pathway. That's not really a target, right? I mean, you can't. Yeah. So it's not. I'm not divulging too much there, but and the fact of the matter is, I've already, for the BMS people watching, I've already received PubD approval, so we're all good. To mention it, but I think that those are things that are really exciting, and I would love to see over time somebody take that up and put it into the clinic, because I have a lot of faith in it. Just based on my experience and what we gleaned from studying that pathway for so long. But there have been a number of programs. I did some key work for apixaban. I don't know if you're aware of apixaban. It's a factor 10A inhibitor. It's an antithrombotic drug. Eliquis is its brand name. So I was asked to, actually, as a physiologist, I was asked to join that team and develop some translational models to understand the antithrombotic potential of factor Xa inhibitors. And I knew nothing of factor Xa or the coagulation cascade, but I learned quickly. And actually, I developed this cool model that I ended up publishing with Alan Tall. He applied it to some of the stuff that he was working on. Who's at Columbia? He's a famous atherosclerosis guy. And but anyway, long story short is I developed an animal model because it was well known at the time that diabetes and obesity were associated with increased propensity for thrombosis. So I took the OB mouse, and just because, again, applying my previous experience in endocrinology to a new thing, and so took OB and DBD mice and showed, yeah, these things have a prothrombotic state if you look at markers of the thrombotic cascade. And then when we then applied the factor Xa inhibitors and showed that we could reduce those risk factors. D-dimer is a classic one, which everybody knows D-dimer now because of COVID, but but D-dimer, with a D-dimer in there, and the fact of the matter is that it actually ended up sort of stimulating a phase 2 trial, so in diabetic patients. So that's kind of impactful. It was in the NDA. I was really excited about that. But that's a situation where it was a breadth of work that on its own really wouldn't stand muster in a published forum. I presented it at a number of different conferences, but I just didn't have time to put it together in a broader package that would have been sort of, suitable for a publication in a good journal. I don't like publishing in lesser journals. I don't. I'm much more of a quality over quantity kind of a guy.
I think a lot of us, I am too, and I think a lot of us are like that as well.
But that's an example. And then I did some other stuff for saxagliptin, which is a DPP4 inhibitor for diabetes. Did some preclinical work on cardiac progenitor cells and stuff like that. I mean, my career has sort of been, if there's an interesting question, I'm going to try to tackle it. And I didn't really care. I've never really cared if I knew the field in and out. So I'm sort of a jack of all trades.
That's exactly what I was going to say. You seem, based on our conversations, that you do have a basic understanding in a field that you studied in, but then the ability to think that you learned while studying in a specific field gave you the ability to learn basically anything and everything that you need to solve a problem.
Yeah, it inhibits your career. I will say that. Taking that approach is not an easy road. Again, it's a common theme of this discussion. It's not an easy road, but the fact of the matter is it's an interesting one. And it provides you with a lot of perspective on physiology in general. So from a very classical scientific approach, I feel like I have a really broad understanding of physiology across many different systems, from neuroscience to reproductive biology, kidney function, cardiac function, metabolic function, and all that stuff. Sort of what you see is it's all just one giant syncytium, and they all work together, and they all have their interplay. And so you can take the commonalities in those different systems and then apply it to almost anything.
Which I think is very valuable in drug discovery and in industry in general, being that, jack of all trades and being able to very quickly understand and apply, knowledge that you've acquired in one system and translate it in a different system, or, putting these puzzle pieces together.
So that's how I actually came across the whole mechanotransduction thing for endothelial cells, actually. So when I was working in bone, I was actually in charge of the in vivo pharmacology for all bone programs at BMS. That's what my original position at BMS was. And so setting up and running bone biology model. And everybody was asked to put together, new targets or whatever. And the best known anabolic bone building agent we know of is mechanical loading, right? So that suggested then that bone somehow responded to mechanical stress by building more bone. So I got into it. I'm like, wow, this is really fascinating, right? And just about the time I was starting up a couple programs and studying how osteocytes responded to mechanical stress, They decided they didn't want to work in bone anymore. So they moved me to cardiovascular disease. And then when I worked on endothelial cells, I'm like, oh, they respond to shear stress. Yeah. I can take what I learned from the osteocytes and apply it to endothelial cells. And that's what I did. So yeah, I don't know if it's necessarily something that applies to academia where you really have to be focused. And maybe I'm wrong. I don't know. It's been a long time since I've been in academia. But it just seems to me that people need, in order to build your academic career, you need to be much more focused on on one or two problems as opposed to being more broad. And I can be wrong, so I don't want to offend anybody, but it's my perception.
I think it depends. I think it depends on the scientist, and it depends on the ability of that person to potentially collaborate. I think you can be very successful if you focus on, I don't know, one protein or one family of proteins, or you can be very successful, as you brought up Michel Bouvier, where for him, because I did speak to him, and I think by the time we publish our recording, his episode will have aired. And I asked him, do you have a favorite GPCR? And the answer is no. And he's always been interested in drug discovery and in better understanding GPCRs in general. And the question was always, how can we, what is the question? What are we looking at? And what are the receptors, or what are the proteins that are implicated in this disease?
Yeah.
And I think, and look,
I can see that, definitely, knowing it.
It's where he comes from. So I think it's kind of you and your luck, or you and the people you meet, and you and the projects you work on in academia. I on the other hand, I think being a basic scientist in an academic setting, at least for me, it was rewarding, but it was difficult. And you mentioned this one of the themes of our discussion today, that it's not simple and it's difficult, is that I only had the energy or the mental capacity to work on one specific topic, which was chemokine receptors.
Yeah.
Do I wish I was able to work on different things? Maybe. But then again, I think it also depends on, so I think you have a limited amount of energy when you wake up in the morning. And if you're focusing on setting up a series of assays to answer one or two questions because you're working quote unquote alone, because you're responsible for the idea, you're responsible for the experiment, then you cannot become the jack of all trades because you're focusing on that little problem. And I think one of the advantages of being a scientist in industry is that the team aspect is different. The teamwork aspect is different. The goal changes. You don't have to, so you have to show results. You have to answer scientific questions and not prove that you can pipette. And I'm really reducing it to the basic, or prove that you can run an assay. I think you have to show that you can think about how to set up that perfect assay to answer that question.
It's about application. And I think that's also, I mean, just from my experience, you see similar types of things in academia, and where you're shifting and you're manipulating your toolbox of capabilities to answer questions that arise in your string of research. I think I see research in academia as more a temporally connected way of doing science, where you're able to sort of follow things along over time. And there's a certain aspect of that to drug discovery, but it ends. It has a defined beginning and it has a defined end. And during in which time you have to come up with a new target if you want to find, or a new application of something. And so and the whole innovation and technology thing in my current job is really what gets me excited. One of the reasons why I took the job was that aspect of innovating in technology and driving it toward doing things that we haven't done before with certain technologies, and how we can sort of move beyond, antibodies.
I know we talked about this as well. So here's my other question. So you're mentioning, that the projects and targets have a specific lifetime or half-life, and there comes a time when, the plug has to be pulled. How did it make you feel the first time, you were working in bone, and then they said, well, done? Well, how did you cope with that?
Well, I didn't cope well. I was a little frustrated, but over time, you realize your frustration is really built around insecurity, and it's your own insecurity with learning something new. And so once you realize that, you're able to conquer it. So, I realized after, a few weeks that really what I was upset about was not so much about not working in bone anymore. It was more about my insecurity and ignorance, if you will, of the new field I was thrust into. And, I got through it. You put your head down and you start to learn, and then you realize that you can learn pretty quickly. Because the one thing you understand, about me in terms of my career path, I was really working in neuroscience and reproductive biology at Wyeth before I came to Bristol to work in bone. And so there was an example of where I had to learn something completely new when I got into osteoporosis. The problem with that was it was basically just a cup of coffee. I spent 2 years working on it. Just at the time I was mastering it and understanding it to where I could really ask some interesting questions. They pulled the rug out, and then you're like, oh my gosh, I got to learn this whole thing new again. The issues with that come with the fact that you're not going to publish papers. You're starting over from scratch. So those kinds of moments do impact your career progression. And if you've done it as many times as I've done it, your progression is pretty slow. But the fact of the matter is that you learn something else. Again, you have to keep a positive outlook and really look at the bright side. You're learning something new, and you have a full breadth of understanding of physiology that perhaps maybe not many people have. And that's the way I like to look at it,
I like that, and I like the fact that you were mentioning that it was hard, and it's a matter of learning something new. And I think as humans in general, we're always afraid of change.
Oh yeah, absolutely.
And it, you're not born with confidence. You build confidence over time.
Mm-hmm.
And yes, it may hurt when somebody pulls out the rug from under you. But at some point, you get to a point where actually you jump up when the next time the rug is being pulled off, because you know what's coming. And
right. Exactly. And you can see it coming, and then you learn how to adapt and quicker next time. I think I learned that when I went into thrombosis, that was a tough thing because it was a relatively drastic step. Osteoporosis is highly endocrine regulated. So that was a comfort space. I didn't really have too hard of a time. But then going into the thrombosis and looking into the coagulation cascade, something I knew nothing about, that was much more drastic of a step. The step into heart failure and endothelial cell function was a little bit less because, again, I was progressing along the same physiological system. So I could, you can take things that you learn with you.
It's so interesting that, we're talking a lot about failure and a lot about, challenges. And I think it's been such a challenging year for everyone in general. So for those of you listening, today is December 30th, 2020. And Brian, you're actually closing the loop of a year of podcasting. The first podcast episode was recorded in February 2020 with Paul Insel.
Yep.
And we're closing the loop, but I think it's although it's been a tough year for everyone on so many levels, being able to, end the year and chat with so many amazing scientists, I think it just made the year just an amazing one. And just because
I think you should be really recognized, because I think it's a great idea. And I think I hope to see more of these kinds of things pop up, because I think it's a really great communication device for especially for young scientists who may not understand or have a resource to go to try to think about different ways of using their scientific knowledge to apply in different areas. So I think, I was really excited when you contacted me, because I think it's just an outstanding concept. So I think you need to be recognized for the concept.
Thank you. I think that's the artist, the scientific artist in me. Yeah. I was trying to think. So we talked about this, about, projects that not everyone may be, it's not mainstream. It's not the flavor of the month. And I was trying to come up with a concept of, still giving back to the GPCR community, still staying close to the science without necessarily staying in the lab. And the first thought I had, it's writing a blog or, writing articles. But then I realized that it's not my forte. I loved writing my papers. It was very difficult, very challenging, because you needed to find the right word. And I think I've heard Michel Bouvier say this, is that, when you write something or you're making a presentation, you want to make sure that you're crystal clear so that people don't even ask themselves questions, because you're just giving them the answer. And I thought the podcast was something that I've never done before. And I thought it was just, let's just try it and see where that goes. And I'm so grateful that today I get emails from people saying, well, we want to hear about adhesion receptors. And I say, yeah, of course, I want to talk about adhesion GPCRs. Right. And get there. So the take-home message when it comes to junior scientists is that if you have an idea, whether it's a scientific idea or anything you want to try, just do it.
Yeah. And I think you're right. I mean, I think there's so much. This is such a better venue than a blog. I'm sorry. I think because this has the personality content that you don't get from a blog with other people, and you get to understand people's personalities and what they're like, and that's always interesting as humans, right? We find that kind of stuff interesting. So I think you hit on the right sort of way to approach it. So yeah, kudos to you.
Thank you.
I hope I answered some of your questions. I don't know. I hope we tend to go off on tangents. Apologize.
I think we might need to do a part 3 of the episode. But I think I always have a lot of fun talking to you, Brian. So I think it's an awesome way of communicating. And we talked a lot about mentorship in the beginning and in the first time we spoke again. And I think one of the take-home messages for people is, just do it. Think about what you want to do. Try it. If it doesn't work, it's okay. It's fine. I didn't know that the podcast would take me where it's taking me. And I never, ever would have imagined being able to speak to so many phenomenal scientists. Just having, the email of Fiona Marshall. I mean, come on. Me working in the lab would have never even, imagined. And she responds and, hey, Yamina, thanks for the podcast. Or, it's just phenomenal.
Yeah, it's just. You're right. You have to be, you can't be afraid, right? Again, you can't be afraid of failure. You can't be afraid, nothing risked, nothing gained, right? You just have to be willing to just put yourself out there. And that's sometimes hard for scientists. Scientists tend to be more introverted. I'm certainly an introvert, although people tell me I'm not. I actually am. But it's putting yourself out there, it's always rewarding in the end. And whether you succeed or fail, you've always learned something, and you've always will take that with you and say, look, at least I tried, right? It's really, really important.
Exactly. And I'm an introvert. Well, actually, it depends. I did a multiple, these personality tests, and depending on when I do the test, I'm either 49 or 51% introvert. So it's a little bit between the two. But when I think back about at the times where I went to conferences and, I saw all these scientists, Nobel Prize laureates, and all these superstar scientists, and I was there alone at my poster, and no one came to see my poster.
We've all been there, Yamina.
And then I said to myself, well, it is what it is. But then I'm glad I still did it. I'm still glad that I stayed at my poster, because I did the work. I tried. I was able to publish the work, whether it's appreciated now or it'll be appreciated in 10 years. I still think it was the right question to go for. And
I think there is this concept of extroverted introverts. I don't know if you've come across this in your reading, but there's certainly a subclass of people who are introverted by nature, but in certain social situations become extroverted. Yep. as a way of sheltering their introversion. So one time I was giving a talk. The first time I talked about this whole concept of receptors as proteins working very similarly. We kind of talked about that last time. I gave this talk at our GPCR conference, and I was so nervous because there was a lot of bigwigs in the room, and I'm putting myself out there in something that could be potentially controversial. Although I didn't think it was controversial at all. And I started my talk off by because everybody before me is like standing up on this stage and they're walking around, talking, giving their talks. I'm like, wow, I could never do that. So I started my talk. I was like, I don't know about you guys, but I'm an introvert, and I'm, this my inside, I'm screaming to stand behind this podium, but I'm going to try to make it, I'll step out from the podium to try to make myself do what you guys did, but there's no way I'm going to walk up and down the stage. It's just, I can't do it. It's just not who I am. It's kind of fun.
It is, and it's interesting. I think and we're completely taking this conversation out of the scientific, well, not necessarily scientific, but the GPCR realm. But I think it's still important for the audience. And I've went through these, and I'm sure, Brian, you did go through these, discovery phases of who you are and what are the things that you're comfortable with in order to get where you are today, and then tell yourself, well, this is who I am. I'm accepting the person I am, and this is how I work. And this is what's important for me.
So, Yamina, I don't know if we were on recording or not. So, remember, we got into this discussion briefly earlier about Jordan Peterson. And
yes, we weren't recording, but I think
right. And so, he in his lectures, he talks a lot about understanding your own personality, and trying to grow so you can grow and develop as a human, and how that applies to what field you choose. And I think it's really important, and we shouldn't discount that, because it is an important part of understanding of where we should be. And and science is no different than that. You have to understand who you are, be confident in who you are. I mean, you are who you are, and own it. I mean,
yep. and
own it, yeah.
And accept it. Exactly.
Yeah, exactly.
And it's sometimes it can be difficult. I think learning who you are, and sometimes it's shocking. You're like, oh, that's me. Oof, I'm not sure how I can handle this. But I think you need to figure out who you are, what you like, what you don't like, which is also very important. And what is your working style, for example, and
that way you find your right path, right? And I do think I push back against the whole concept of behaving in a scientific way, because at least when I was being trained, there was a certain way you were supposed to carry yourself. There were certain words you were supposed to choose.
Cool.
Yeah, no, but really that's the way the training was. It's like, they trained you have to present yourself like this in order to be taken seriously as a scientist. And I've spent 25 years pushing back against that, because I don't think that's really accurate. And I feel like as scientists, we should be who we are. Just because I don't act like somebody else doesn't mean I'm not a good scientist. It just means I'm different. And, so anyway.
Exactly. No, I was just going to say that, and once you accept who you are as a scientist, as a human, and you're at peace with the person you are, people will see it, because that'll also show how confident you are at the end of the day.
And we need that, right?
Yeah.
This whole concept of diversity and of thought, diversity of background, and so forth is really, very important to innovation and driving the next discoveries. So if we're all sort of like everybody else, we're not going to innovate.
Exactly. So whether you're an introvert or an extrovert listening, it's okay. Both are okay. Just do your thing, follow your own path, and then
be true to yourself, right?
Yeah. I think that's very important. So one last thought. I did this insight test to figure out, kind of my work style and things like that. And it's interesting because I can be very scientific, but I can be also very emotional at the same time. And apparently it's very rare having, that straight logical thought and the emotion associated with it. But my husband could attest to the fact that in the lab, my bench looked pristine, and at home, my desk did not look that pristine. So then again, we all have our little issues there, but I accept the fact that my desk doesn't look as pristine as my bench used to.
Mine doesn't either. So there's two of it.
I'm pretty sure it's not just the two of us at this point, but it's kind of it's who you are, and that's it. As long as you find what you're looking for in that environment, then you should be good. All right, Brian. I think we went around, we talked about your career, how industry works, what do you do, how you think about some of the problems that you work on, and how you cope with the fact that, some projects get cut because it's no longer a valuable route to go for. The last thing I wanted to kind of touch upon is we talked about how projects get chosen. And I think one thing that we didn't really articulate is the fact that there is so many things that we all could be working on. And the fact that at some point you have to make a choice.
Correct.
On that. And I was wondering, what are your thoughts on, making that choice, or how do you?
Wow, that's a really highly personal thing, right? I mean, that's really comes down to your own personal preference and intuition. Intuition should never be discounted either in scientific endeavor. There's a huge component of intuition. And I've always relied really heavily on my gut. Not to say that data doesn't drive decisions, because they do. But when it came to generating hypotheses, there have been times in my career when I just knew that was the right answer. The whole thing with the FSH story, that was all based on intuition, because there was no existing data. We had to generate it. So it's, when you're really innovating, intuition is really, really important to generating hypotheses. And then you use the data to drive the decision, right? So the hypothesis is driven, I think, from intuition, at least in my case.
Yeah.
Based upon historical data as well. That's not to say I don't read the literature, because I do. But the fact of the matter is, it has to make sense. But there's certain all the key discoveries I've made in my career were all based initially on intuition. And then, and how the puzzle pieces fit together. And then from there, you use the data to drive whether or not you're correct. And there's been plenty of cases where I was wrong. But it's more than half the time I'm right.
which is already pretty good when you think about it.
It's pretty good. I think it's pretty good. I think that's all you can hope for. But the important thing is to be confident enough in yourself to say, no, I was wrong. And that's really another important concept, right?
I think
people, you have to have confidence in your ability to say, no, I was totally wrong.
Yeah, I think that also comes with confidence, as you mentioned, where sometimes you're wrong. You cannot always be right, and when you're wrong, you're wrong. But when you're right, it's all the more, you savor it even
Absolutely.
more.
I'm still savoring it. That whole circadian stuff I did back in the late 1980s, I'm still savoring it. Because that's what we are as scientists, right? We want to have an impact, and we want people to recognize it. And so when you do have those brief moments of success, you definitely will savor it and never forget it. Some people are better at it than others. Certainly there are people that are a lot better at it than I am. But I think that, again, you just have to have confidence in who you are and your abilities. And that's not easy in this field, because we are wrong a lot of the time.
Exactly. But that's the beauty of it. And I think that's why we need a diverse crowd, and we don't need scientists to come out of the same molds and act the same way. The more diverse we are, the more, statistically, the more answers we get together.
That's right.
Correctly.
Absolutely. And again, this comes with collaboration. This comes with cooperation and working across. And I seeing in my career over time, the amount of cooperation and collaboration that now takes place compared to when I first started, is very, very encouraging and heartening to me, because I think there was a time when scientists were very sort of isolationist, at least in the way I grew up in the field. And now to see people working across many different universities and working on problems in a more global way, I think is just awesome, because that's how we're going to really solve the tough problems.
I agree. And it's for a long time, I thought I was was kind of naive of thinking that, why all the secrecy, and why don't we work together? Because it's so much faster and so much better. And I think it's a more harmonious way of getting to those answers. But now I see the difference as well.
I think this is why it's important to choose your collaborators carefully. But you can't be totally naive, but because it is somewhat of an idealistic viewpoint. It's always based in reality, but I think it's more and more based on reality. And the more especially I'll have to say what I've seen in pharmacology lately is really, really positive. Seeing people coming together to work on problems and across areas, and so it's really cool.
It's also one last thought about collaboration. I think it's also a matter of compatibility. As when you say choose your collaborators, you will choose your collaborators based on your personality and their personality.
Mm-hmm.
And it's like it's like choosing your friends basically, and you're doing something
totally,
together.
You're totally right. Totally, it is. Choose your friends carefully. You have to be careful, but at the same point, I think if you're a good scientist, that will come out in the wash. It will always reveal itself in the end. Even though you may go through challenging times or whatever, if you stay true to your principles and your ethics and your morals and who you are, it'll ultimately reveal itself.
Absolutely. So this was a really great pleasure of chatting with you. I think we went from science to philosophy through Jordan B. Peterson, but I love these conversations. Thank you so much, Brian.
No, thank you again for the opportunity. It's just awesome. For somebody like me in industry who, works in anonymity most of the time, it's really, really a rare opportunity, and I'm very, very grateful for it. So thank you, Yamina.
Absolutely. Thanks so much, Brian.
All right. Take care.
Bye-bye.
Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, and Jin Chung. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcasts. Another way to support us is to share the podcast, the newsletter, and the virtual cafe with your colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.